So essentially we're going to make a standard, right? We're just going to make one standard, and that standard, if I'm not sure exactly what volume we want to be able to make, and we don't need to make it in a volumetric flask necessarily. But if we make one standard that contains all of our ions, and in that email I said the concentrations. I can't remember them right now. Do you want to? Let me look up that email real quick here.
I have it pulled up over here.
Okay. So it's going to be this one solution here is going to be 10 parts per million calcium, 5 magnesium, 0.1 iron, 0.1 copper, 0.025 aluminum. Is that what you saw too? Okay. And to make that, if this volume was going to be 400 milliliters, then you would put in 4 milliliters of 1,000 parts per million calcium. All right. And then you would have 2 milliliters of the 1,000 parts per million magnesium. Now I say the 1,000 parts per million because that's how they usually come. Okay. The standard solutions usually are about thousands. They come in 1,000 parts per million. That's their concentration. Okay. So let's finish up the recipe here. 40 microliters of 1,000 parts per million iron, 40 microliters, 1,000 parts per million copper, 4.3 thousand parts per million aluminum. Okay, so if you can find all of those, then you would add. You want to fill it up to 400. So that's 392 milliliters of the 2% nitric acid. Now, did you get to pass by the lab recently?
No, not since you've sent these numbers.
Okay. So I think that across from the mass spec, do you remember which one the mass spec is?
I do not know.
Okay. Did you get to go with Meow into the and to see the ICP or no?
Yes.
Okay.
So is it in that same room?
Yes, that same room. And if you're facing the ICP and you step back about 10 steps to your left, there's that black bench top.
Okay.
It should be on that bench right there to your left.
Okay.
If not, we can just make some new 2% nitric acid, or if we don't have enough. But we don't need 400 mils. I was just this is a model for a 400 mil sample. If you wanted to only make, the problem is we can't get this value much lower. 4.3 microliters is already pretty low. So
okay, yes.
if you
400 milliliters would probably be the lowest we want to take it.
Yeah.
Okay.
But we should be okay if we use this as a standard and just keep it. If we made 400 mils, that should probably be enough forever. And we can just kind of store it. And then when we're ready to use it, we do what's called a serial dilution, right? We just take...
You were talking about at the beginning of the email, right? Or towards the end?
Yeah.
Okay.
Was that clear enough? Mm-hmm. Okay. Yeah. You just add an equivalent amount of water to this, right? So 1 part. And it's usually going to be like 10 milliliters. 10 milliliters of our standard. So this is standard 1. And then 10 milliliters of water. No, sorry, not water, but the 2% nitric acid.
Okay.
And then that'll give you standard 2. And then you do this same thing. 10 milliliters standard 2, and then 10 milliliters of your solvent, nitric acid. Right? And that would give you 3. Okay? So all the concentrations would go down, and then you would have your parts per million of these different things decreasing down as well. And then we'll just have our blank will be just nitric acid. Okay. Now I don't know if our machine can detect these concentrations of iron.
Okay.
So Ben Lee's waiting. You want to admit him, or I guess I can admit him. Did that work? Do you see him?
Yeah, I see him.
His picture?
Mm-hmm.
Oh, good.
Are you there, Ben?
Yeah.
Okay, perfect.
Okay. All right, good. All right. So, yeah, so that's how we're going to make these standard concentrations. I don't know if our concentration of iron that we make is going to be able to be, or the concentration of iron that we have in our media is going to be able to be recognized. The bovine serum albumin might have a little bit, but I don't imagine it'll have much of anything.
So it'll still be pretty low.
I think so. Another thing that is kind of a problem is the other counter ions in the ingredients in the serum, or not the serum, but the media, like had phosphates, and there was one other one. I can't remember if it was sulfates. Yeah, sulfates. So sulfates are less insoluble, but phosphates can be pretty insoluble. So iron phosphate and what are other copper phosphate? I would expect those to precipitate out. So, but they had iron in there, and they had phosphate in there, and they didn't get precipitation. It's probably because their concentration is low enough below the solubility, because all insoluble compounds are soluble to some degree, right? I don't know if you remember that from 106, but so that could be the case. I don't know. It's hard to say, but that's all right. Whatever our standard ends up doing for us is fine. Really, what we want to kind of do with this is we're going to be spiking our media with concentrations of these ions, right? Concentrations of iron, copper, and aluminum. And then we want to grow the cells in those spiked samples. But we also want to be able to, after we do the spiking, take a small sample of that, go run it over on that machine, and just verify that its concentration of our whatever ion we're interested in is what we think it's supposed to be. Does that make sense?
Okay. Is that where some of the numbers of like the decreasing by half comes into play? Where we have like the 10, 5, 2.5, 1, 0?
In terms of what?
In the email when we're talking about like dilutions, you would say that like the standard would have, like for example, calcium would be 10, and then 5, and then 2.5, and 1.25. Is that where you talk about like decreasing it so you get like
A standard curve.
okay?
Yeah. So on the ICP, you give it your standards. So you say, okay, this standard has, so their first standard is going to be 10 parts per million calcium, 5 parts per million magnesium, blah, and so forth, right? And then you have the second standard. The second standard is going to have half of these, right? 5 parts per million, 2.5, 0.05, 0.05, 0.00125. And then the third is going to have half of that. The fourth is going to have half of that. And so what that's going to do is we're going to be able to look at any one of these ions, and it's going to have a signal, right, absorption, and we're going to be able to plot our concentration of iron versus that signal and get a standard curve. And then we're going to run our media, and our media is going to, the media should have, what was the concentration that I calculated? It was like the media, so the first line there, it says that looks like our media already has 72 parts per million calcium, 0.0138 parts per million iron. So 0.0138, and our low on our iron is going to be all the way down to 0.00, no, that's for 0.0125. So this is going to be 0.0125, the low, right? And so we would expect our media to give us an iron concentration right about there. Now, Oh, I did those calculations somewhere. Oh, here it is. Well, I just realized the problem here. So I'm looking at this email here. This is the original concentration of the iron in the media, right? And the problem is when you run something on the ICP, you want it to be mostly in your 2% nitric acid, right? So I think I said in here somewhere that to be sure the samples of ions are soluble, you always will take like 1 milliliter of it, of the media, and add 9 milliliters of nitric acid. Does that make sense? So if we do that, we're going to basically see 0.00138 iron, and that's outside of the range for our lowest iron concentration.
Okay, that's why I was confused. Down at the email it says 0.022 iron. I just didn't get where that number came from.
Which one? Which one, Ben? 0.022?
Yeah, 0.022.
Where did I say that one?
ppm, that's just down, kind of like the middle of the email. So like the paragraph that starts with because of this dilation we can expect.
Oh, okay, yeah. Well, what I'm referring to there, in this little section here, is this is going to be the dilution of our spiked sample, right? Because we want our spiked media is going to be about 4 micromolar metal ions. This will give us 2 ions for every peptide that we have in there, right? Am I seeing that right? Is that right? Because we dealt with 2 micromolar peptide at the highest concentration.
Correct.
Okay, so 4 micromolar ion. So we want 2 times the beta amyloid concentration. So we'll have 4 micromolar ion, metal ion. And 4 micromolar metal ion, I think that was, let me see, I did that here. Yeah, 4 micromolar metal or iron ion. I don't know if I said that on here anywhere. I didn't. But the 4 micromolar metal, 4 micromolar iron. So 4 micromolar iron is 0.223 parts per million iron. And 4 micromolar copper is 0.254 parts per million copper. So if we did a 1 to 10 dilution on that, that's where we would expect to see, we're looking for the 0.02. And that 0.02 should be within our dilution range for the iron, I think, right? So iron goes all the way down to 0.0125. Yeah. And the copper. Does that make sense, Ben?
Yeah.
Okay. All right. So, yeah, this is the goal. We run an experiment where we have a control. We're just growing cells, right? And we see how happy they are. We do the same thing. Well, okay, now remind me again how you guys did the peptide before, Trevor?
How we...
You would put the peptide in there and then see how well it grew, correct? You wouldn't...
Correct, yeah. So
okay.
we put the peptide in with the cells for 48 hours, and then we measured the number of cells at the end of that 48-hour period.
Okay, great. So we're going to do the exact same thing without peptide, right? And then we're going to do the same thing with peptide. And then we're going to do the same thing with peptide and iron. And then we have to do the same thing also with just iron. That's going to be another control to see if there's something wrong with the iron.
Okay.
And we should probably do that one with the control of so do your controls first. Positive control, meaning we expect plenty of growth. So that's just no peptide. And then your other positive control is, no peptide, no iron. And then your second one is no peptide with iron, right? Those are both cheap, right? If the iron just kills everything, then we don't have to worry about looking at peptide and iron, right? Does that make sense? If the iron kills everything, we try to lower our iron concentration until we get some growth. We could also do no peptide with copper, right? And see if the presence of the copper ion kills anything. But that email you sent me about the link in there about the bovine serum albumin, not the bovine. Is it called bovine serum? Fetal
bovine serum.
Yeah. This is kind of interesting and kind of concerned me a little bit because it says FBS remains a popular supplement because it provides a wide array of functions, delivers nutrients, growth, attachment factors, and protects cells from oxidative damage. And our hypothesis with how the peptide is killing the cells or preventing the cells from growth is basically we're suggesting that it's an oxidative form of oxidative damage. Because basically what we're saying, or this is what we're trying to test right now, is if that peptide, right, somehow we have a feeling that it's interacting. Well, I don't know if the reviewers agree. We'll see, but we have a feeling that it's interacting with these cells and preventing them from growing. And how is it doing it? Well, we've looked up and we've done some research, and we think that it's possible that it's getting metal ions and then coordinating those metal ions to the peptide, and it makes itself a little bit of a kind of a catalyst for oxidizing species, kind of like myoglobin or hemoglobin, right? That iron there can go from iron 2 to iron 3 really easy if it's kind of coated and covered. And if it comes along anything and just pulls an electron away, oxidizes that surface, that can be detrimental to a cell and kill it. So that's what we're trying to find out. Does our effectiveness of our peptide, is it affected by the metal ion? Because if it is, that would suggest that our metal ion is interacting with the peptide in a way that helps it do its thing, right? We could also do some spectroscopy and just see if the absorption peak of the metal ion, because if you look at just the absorption spectra in a visible UV vis, right? Just look in the visible range, you're going to see this sort of a thing with iron. Iron looks like that in solution. And if we add our peptide, and we can see if there's a slight change, because what that slight change would mean is that the peptide is actually binding the iron and changing how the electrons are kind of moving around the iron. And if that's the case, that would be strong evidence that we're getting some sort of a complex. So if we see that this iron complex actually does do something supplementive to our peptide, then we look at this as a backup to try to see if, further evidence, right? It'd be really, really, really interesting, in my opinion. No word back from them yet, huh?
No, so like I think I forwarded you the one that I got back. I think it was last week or about 5 or 6 days ago. And I think that what happened is because they changed over during the summer, I think they stop reviewing, and I don't think they're going to start until their classes start back up again.
Okay.
But the indication was positive that the new contact that she put me with is going to start the review process and is going to be emailing us at some point with the review notes from the reviewers.
Mm-hmm.
We'll see what feedback we get from them.
Yeah. That's a good thing to keep in mind. But it's kind of a shame as well because it kind of ties our hands for a long time, whatever. But it's okay. This would still, if they came back and said, yeah, we don't feel like it's indicative of anything, if we did have evidence of this sort of thing actually contributing, then it would just strengthen the argument. We could just add this right onto it and do some of this as well and have some good evidence. Okay, so I think we want to start with no peptide, no iron. That's just control, nothing, right? And then no peptide with iron. And then no peptide maybe with copper. If we do those controls and just see if the iron and copper influence the growth.
Okay.
What do you guys think?
Sorry, I just had a question. Where does the peptide come from?
The peptide, we buy it and we just add it into the media.
Okay, so the peptide is just what you guys have been using to see how it changes the cell growth.
That's right. It's the same peptide that is in the Alzheimer's brain tissue samples of Alzheimer's patients, right? It's the protein that seems to be overexpressed in Alzheimer's, in CTE. Is that true too, Trevor, or no?
I believe so.
Other forms of dementia, right? I'm not sure which ones, but so whenever there's we should look into that a little bit more too, because if it is associated with just trauma in general, that would be also another kind of an immune response perspective. But what was that latest article that I saw that I sent you?
Was that the one with the vaccination and the prevalence of Alzheimer's?
Yeah. What was it? If you were... Oh, no. That was the one, I think it was... maybe it was... What was that one? I can't remember. What was that one?
Yeah, so that one was, they were showing that it was kind of it was not what they were expecting, because they were expecting that when you have the vaccine and then you have an increased immune response to that, then you would see an increase in the presence of Alzheimer's, but they saw the reverse. That when you have the vaccine and you have that known immune response, you have a decrease in the prevalence of Alzheimer's.
I'll have to look back at that one again. But then the other one was how if you don't brush your teeth, you increase your likelihood of getting Alzheimer's. Remember that one? Did you see that one?
Yeah, the one with the gingivitis.
Yeah.
Yeah.
Which is, just kind of strengthens the idea that it's an immune response, right? When your body is dealing with something overloaded, right?
Correct.
Anyway, I wonder if there's this relationship between immune response or Alzheimer's and diabetes. We should look into that and see what. Have you ever seen anything like that?
I haven't, no, but I'll put it on I'll add it to my list of things to look into.
Because diabetes, either way, whatever the answer is, it would help us. Diabetes kind of is like an autoimmune disorder where your body is so overwhelmed with generally too much weight. I'm type 2 diabetes, right? That it just can't respond well. Did you hear that? Vaccines generally don't work with people who are obese. I didn't know that, and nobody really has to care about that because generally we're vaccinated against relatively rare diseases. So you're vaccinated as a kid, but they're saying that if you are vaccinated with some COVID-19 vaccine, it's not going to work on you if you're obese, right? And so essentially they're saying your immune system is just already on full throttle. It's not going to be able to respond to things like that. And that's also consistent with why people who are dying from, why obesity is such a significant risk factor in the COVID-19 situation, right? So, I mean, it'd be interesting to see if there's what the relationship is. I'm sure there is one, whether it's less or more, I don't know. But what's the relationship between Alzheimer's and diabetes?
Yeah, I'll look into that. That's interesting.
Okay. Anything else we need to talk about?
So Ben and I we can start to put that standard together. We can look through the lab. I'm pretty sure we saw the nitric acid, and then we can look for the other solutions. Is there a particular spot that you think we might be able to find more of them?
Most of the metal solutions, the 1,000 part per million solutions, right? Most of these are in, so aluminum is the only one that I don't know if we have, but everything else should be on campus for sure. And the increased likelihood, the most likely place is the furthest left cabinet above the ICP.
Okay.
So you know where the computer for the ICP is?
Correct.
And there's a cabinet, a brown cabinet up above. And then if you follow that all the way down to the end of the room, that far left cabinet, it's far left 2 or 3 actually, have all the samples of metal ions for ICP. And you want to make sure you don't cross-contaminate those samples with each other, right? Obviously. But other people use them for other things. Yeah, I think there's nobody in the stockroom, right?
No, I don't think so.
If it comes time that you're there and you can't find it, give me a heads up saying we're heading to the lab, or we're going to work on it right now, or whenever it is, so that I can be by my computer and we can conference in really quick, and I can lead you to them if we can't find them. Does that make sense? Okay.
So just one last question.
Yeah.
Because you were talking about how the iron concentration, like 0.0138 ppm, is not within the range. Would that still be a problem?
Well, it just basically is saying that when we run the 1 to 10 dilution of our media, we're going to be able to see the presence of calcium, magnesium, and that's it. We won't be able to see iron and copper and aluminum, right? But that's all right. All that will do is just tell us that our method is working. You could make a spiked sample of the iron, try to make it 4 micromolar, for example. Now, I wouldn't make it 4 micromolar with the 2% nitric acid. Well, you could do that. It wouldn't matter just for your control, but that wouldn't be the specific one that you'd actually use for your sample. You're growing your cells, right? Because you don't want to put the nitric acid in there. When you end up spiking the media, it might be okay. But because there is some buffer in there, right? But 2% nitric acid is kind of a lot. I don't know. If not, we'd have to make ourselves some spike standards, right? So some concentrations of iron and some concentration of copper and aluminum that we're going to be using to spike it with.
Okay. Right now we just start with the 4 standard.
We're going to make the standard, and we're going to dilute it into create 4 different standards. Let's see, 4.
With the 0 and 5.
Yeah, that's right. Okay, good. And then with the 5th is 0, and then those are going to be what's going to give us our standard curve. And then we'll run our sample, and we'll see where. So you're going to get not just one of these for iron. You're going to get 5 of these, or not 5. 1, 2, 3, 4, 5. Yeah, 5. You're going to get 1 for each element, right? So you're going to get 1 from aluminum, 1 for copper, 1 for magnesium, one for calcium, right? And they're all going to have their own slopes, and you're going to find out what the concentration of these elements in your sample is, right? It's just media, right? So you just take some media and run it 1 to 10 dilution with nitric acid. And then you could take some media, put a little bit of iron in it, and then run it again and see if it shows up where you think it's going to show up. So we're looking for a couple things. There's a possibility that the media kind of masks the signal, that there's something in the media that's causing it to either precipitate out or just absorb differently. And that's all right. We just got to know what kind of signal response we expect to see. Yeah. And I would be interested in also running your samples after they're done. Let's see. Is the process destructive? Does it take the solution? It does, doesn't it? You can't get the solution back after you're done, right? So if you had well, and it takes all 300 microliters or what?
Yes, it does. But we could run it to where we have, for example, if we have 20 different wells,
right,
we could measure 10 of them for the cell counts, and then we could pick from the other ones to measure the ion concentration, if that's what you're thinking of.
Yeah, yeah. Something like that would be good, I think, because then we just kind of get a handle on how is the iron concentration changing as this process proceeds. Because if they're just consuming the ions for whatever reason, I don't know why they would be, and the ions aren't available to kind of complex with the peptide, then that would be it wouldn't be working the way we think it's going to work, right? And so like if we had some, so like you said, we have lots of wells. Some are going to be growing, and we're going to use them to count, right? But then 1 or 2, or however many, we're going to use to check the iron. Well, this stuff right here. Now, when you count, do you put some sort of trypsin in first to get the cells off the ground?
Yeah, so we have to put the trypsin in first to remove the cells from the plates, and then we transfer them into cell cups.
Okay.
And they're counted from there.
Okay. So you would do the same thing here, obviously, right? You want to get all the cells out. But it would be interesting to put into a centrifuge, one of your mini centrifuges, and that way you would separate. If you spun it at fast speed for about 10 minutes, all the cells should be stuck down here, but all the iron should still be in here and in the supernate. And if you did that with a different one, but didn't centrifuge it, then your cells and your iron should all be in the same mixture. And so we could compare and see where the iron's going. Is the iron sticking with the cells, or is it just staying in solution? Does that make sense? So when we would take this sample here, we would just take off the top little bit here, and dilute it down, and then run it on the ICP.
Okay.
Just an idea, but yeah, having all these things available to us, all these different samples and possible experiments is good. So what classes are you guys taking? Are you guys in my analytical class at all?
I am not.
Are you, Ben?
No, I'm not.
Oh, good.
Biology major, so it's not required.
Yeah, that's good. That class is not going to be good. It's just crazy. I mean, because usually we're just on the instruments all day long. And
Yeah, I was talking to my sister about that. Some of the laboratory courses, I have no idea how they're going to be online.
yeah, well, it's just going to be the best that they can do. They've had online labs for a long time because some universities, well, not really universities, I don't know. Some people use them as intro material, which, after I've kind of interacted with them this summer, I think I might use them as intro material. They kind of cost money, right? So it's like $50 a student. The university is going to cover that, but Yeah, you kind of have to weigh that against using them as intro material just so that students can kind of be a little more familiar. You already write a bunch of pre-lab and do this and they don't read that, so why would they do the other stuff anyway? So anyway, but yeah, it's been out there for a long time, and whether it's going to be, well, clearly it's not as helpful. I mean, you know, the majority of people know that the kinds of things you learn in lab are not the kinds of things that you can just put on paper or in a video. So hopefully it doesn't last long, but man, Hawaii is not going to respond. Is you know, it's going to go like that for, it could go for 3 years, right? I mean, I don't know what their end game is. They have no end game. They have no goal of, they have no idea that, okay, we'll do this for this amount of time, and then we'll find ourselves in a situation where we'll be okay. They have no end game. They have nothing. It's just like they're behaving like a very old retired person, and just saying, keep everybody out, stay out of my house. I gotta be safe. You know, this, I don't know. They're just like kind of actually already pretending, I'm gonna die in a few years anyways. Just stay away from me. It's just crazy.
But some news say like the vaccines should be coming out like maybe.
Can you close that door for me, Tom? Yeah, no, that is a possible end game for them, is that they're going to get this vaccine and then everybody's going to take the vaccine, right? And yeah, that's a possibility. Yeah, who knows? Hopefully that happens sooner than later. People are talking, I mean, the Russians already say they have one. Maybe it'll start showing up here under the black market. That'd be awesome.
Black market Russian vaccines, that'd be anyone.
Well, I mean, just 5 people take it. Yeah, I'm still alive, you know. Did you ever see the movie? Well, I can't remember the movie. But I mean, that's an easy way to win $1 million. Not that this is what's happening, but if you make a disease or make up a disease and then make a vaccine for it and say, I'm selling it really cheap, just $2. But man, you do not want to miss out because you're going to die if you don't get this. This is a bad disease, you know. Studies are showing, I mean, I don't know, have you guys been keeping up on this? It seems like a high percentage of people have zero symptoms, right? I mean, is that what you're seeing?
Yeah, I'm seeing that most of the people that test positive end up not being symptomatic.
Yeah. I mean, they tested huge amounts in London just as it was a good experiment, which we should have done a long time ago. Just test a bunch of people. And I think they were at 80% asymptomatic, right? So these are people who are just random people. And the people who tested positive, they didn't know that they ever had the virus or anything like that. They're testing positive for antibodies, right? So, yeah, that you know, if you wanted to say, I have a secret Russian vaccine, and you sold it to a lot of people,
Good way to make a lot of profit.
yeah, people would just say, yeah, I'm healthy. I didn't get anything, nothing. Oh, that was the movie. It was after World War II. It's a true story, actually. After World War II, the mobs were getting antibiotics, and they were diluting them down, and they were selling them to people. So you get 1 bottle of antibiotics, and you sell it to 10 people for the same price. And, you know, it would work for some, it wouldn't work for others. And, oh, sorry, you know, it doesn't always work. It's just a medicine. But they were making lots of money that way until the police started cracking down on them. So anyway, anything else?
No, I don't think so. Is there a way that you can send that recording to Ben and I so we can have this reference?
Yeah, I'll put it on YouTube. Is that okay? I don't think anybody's going to be searching it, so your privacy will be protected. Okay. Let me know when you guys start doing whatever you're doing. Not that you'll need me, but if you do need me, then I'll give me an increased likelihood of being able to be near a phone or something. Okay?
Okay, perfect. Yeah, Ben and I will talk this afternoon. We'll figure out some plans for the rest of this week and this weekend of when we're going to start some of this stuff.